So this is a lecture on CRISPR-Cas systems, both in their natural context in bacteria, as well as in sort of more laboratory context in their function in genome editing. And so what the objectives for today are to just talk about what a CRISPR-Cas system is in bacteria, talk about what its function is in bacteria, and how it exerts this function, as well as talk about CRISPR-Cas9 systems, how they originated, and then how they were adapted to edit genomes in eukaryotic organisms. And finally, to talk a little bit about the considerations that we have to make when talking about gene editing, especially in humans. And so a CRISPR-Cas system is a bacterial defense mechanism that is used against bacteriophage or viruses. And it's similar to an adaptive immunity for bacteria. So one defense mechanism that bacteria have adapted is antibiotics, right, to kill competitors around them. And we have also commandeered antibiotics for our own personal use. And in the same way, this bacterial defense mechanism against viruses, or this CRISPR system, is something we have commandeered and used for our own applications as well, which I'll talk about later. And so what CRISPR stands for is clustered, regularly interspaced, short palindromic repeats. And you can see these repeats here in this blue section. And so to break this down, right, they're clustered in a small area of the genome. They're regularly interspaced, which means they've got spacers in between each of the repeats. They're short in size, and they're palindromic, which means the sequence of nucleotides in each of these repeats is the same backwards and forwards. And this series of repeats and spacers is what's known as a CRISPR system. So this whole area here is considered a CRISPR system. And what's important to know about the repeats is that the sequence in each of these repeats in a single CRISPR system is identical to one another. Right? So the repeats in the CRISPR system are all the same. However, when we look at the spacers, or those lines in between the blue blocks, we can what scientists noticed when they first started to study CRISPR systems is that the nucleotide sequence in each spacer is different. So while the repeats are identical, the spacers are all different and diverse in sequence. And when they started to look into what these sequences were, what they noticed is that the sequences in the spacers are really similar to those of viral genomes. And so what the spacers actually do is give you a history of the viral infections in a bacteria. And so what seems to happen is there's a new spacer and repeat integrated into the CRISPR system every time a bacteria is infected by a new virus. Some of that viral genome makes up the spacer, and then the repeat is added behind it. And so the first viral infection encountered by that bacteria would have made up this first repeat and spacer combo. And every additional viral infection encountered after that would account for the new repeat and spacer, new repeat and spacer, because these new spacers are inserted here in between the Cas and CRISPR parts of the system. And so when you look at a CRISPR system, you can sort of see a whole history of all the viral infections that a bacteria has had, right? With the oldest ones being seen in the spacers here and the newest ones here, because every new infection is added into the center as another repeat and spacer from that viral genome. And so the longer the CRISPR system is, the more viral infections that bacteria has had. And so if a bacteria has had more viral infections, you would expect it to have a longer CRISPR system or a larger one than a bacteria that has had less infections. And not to be ignored, but the Cas portion of the CRISPR-Cas system is also necessary for exerting this immune function. And so these Cas genes vary pretty widely. Most of them encode enzymes that are function in the immune aspect of the CRISPR-Cas9 system. And so how does a CRISPR system work to be a defense mechanism against viruses? How does it do this in bacteria? It does it in a series of 2 steps, the first of which is adaptation. And an adaptation happens when a virus infects a cell for the first time. And so once that virus infects the cell and a cell survives this viral infection, it can add a small piece of the viral genome, in this case this green chunk here, into its CRISPR region in the spacer, right? And so this cell will continue on living. It's now got a little bit of this viral genome in its CRISPR system, so that the next time or the second time a bacterial cell encounters the same virus, it can undergo the second stage of protection known as interference. And so when a virus infects the bacterial cell for the second time, this viral sequence can be transcribed into mRNA. And then the Cas proteins or Cas genes that are part of the system chop up this piece of RNA into smaller chunks known as cRNAs or CRISPR RNAs. The Cas proteins then make complexes by binding to cRNAs and guiding them to viral genomes and then degrading them. So the RNA guides the Cas proteins to this genome target, and then the Cas proteins degrade it. And when you degrade the viral genome, it can no longer replicate, and thus infection is prevented. And so what that looks like in an animated view, you can see here this bacteriophage inserting its genome for the first time. Little bits of this viral genome are then inserted into the CRISPR regions of the bacterial chromosome. RNAs are made from that, and the Cas proteins can bind to those cRNAs and wait around for the next time that a virus tries to infect the cell. And now all of those viral genes are degraded and an infection is prevented. Right, so let's watch it again. Here's the adaptation phase. The viral pieces getting integrated into the CRISPR spacers, making of cRNAs, the binding of the Cas proteins that hang around and can be targeted to the viral genome and degraded up. And so how this works in terms of editing full genomes in the laboratory is by using a specific Cas protein that can work all on its own, known as Cas9. So it's one of the Cas9 or Cas proteins encoded by bacteria. It's an endonuclease for DNA, which means it's an enzyme that chews up DNA and makes double-stranded breaks. It's guided to the correct place of these, or guided to wherever it's going to break the DNA by RNA. And so using a guide RNA, Cas9 can be directed to a particular place in the genome and cut that particular area. And so the way that a guide RNA is designed is to contain a complementary sequence to the target, which you can see in orange here, as well as a specific piece of RNA that can bind to the enzyme Cas9, which you can see in blue. And so a guide RNA will be designed to have this Cas9 binding portion as well as a mRNA target portion. And so since the guide RNAs can bind to both the DNA that you're interested in editing and the enzyme, it's a good way to ensure that there's some specificity of where Cas9 is going in the genome and where it is cutting to ultimately edit that genome. And so once the guide RNA has found the correct target and brings Cas9 there, Cas9 is an endonuclease, so it can cut the DNA in this very precise location, which will allow for the insertion or deletion or modification of DNA at that particular site, depending on what you're interested in doing. And so in some cases, if you're interested in inserting a new piece of DNA here, along with the Cas9 and the guide RNA, you would include that DNA of interest, and then the cell can use its own repair machinery to fix the breaks that Cas9 generated and insert your new piece of DNA. And so ultimately, editing genomes with CRISPR-Cas9 is similar to either a copy and paste kind of function in a Word document, or what I kind of find more similar is a find and replace, where the guide RNA helps Cas9 find all the genes that you want to edit, and then Cas9 cuts them, and the cell's own machinery replaces them. And it really is extremely simple, right? And it can be used to cure a lot of diseases, especially ones caused by one particular gene, such as sickle cell. And so in this example here, if a patient has sickle cell disease and it's caused by this DNA mutation, we can use CRISPR and generate a guide RNA that targets this particular portion of DNA, mix that guide RNA with the enzyme Cas9, and then the patient will get an injection of this guide RNA Cas9 mixture. And then the guide RNA will guide Cas9 to the affected sequence. Cas9 can cut it out, and the piece that you would like to replace it with can be inserted here. And now the patient no longer has a mutation and no longer has disease. And this can work really well for diseases that are caused by one gene, but you can imagine that it would be more difficult to edit a genome of a patient that has a disease caused by multiple genes. And so sickle cell is one case where this works well. Huntington's disease, some early onset Alzheimer's would be some other possible targets. But this process is amazing, right? It's really elegant, it's really simple, and you can cure a lot of genetic diseases using CRISPR-Cas9 genome editing. But there are some things to think about. And as a scientist, sometimes I just feel this need to be like, yes, let's edit all these things and fix them. But you have to think about some of the practical considerations, like the fact that you might target the wrong gene and edit something unintentionally and totally mess up an organism. You can also have unintended consequences for future generations. So if you edit an animal to be unable to pass on a virus or act as a vector, it's possible that by editing an animal in kind of an unrelated way, you could throw off a whole ecosystem. Additionally, there are the moral considerations associated with genome editing. There's this idea that editing a genome is playing God, and it's not up to us to make these decisions. There's also the idea that not only can we edit genomes of people with genetic diseases, but we can start to edit sort of unrelated, unimportant things like eye color or hair color and create designer babies. And that could ultimately lead to eugenics. There's also this idea of regulation and deciding who can actually regulate this process, right? So is it the government? Is it the scientists? Is it the parents or the patients? And trying to decide how to regulate and control something that's happens so easily you can actually kind of probably do it in your garage is a really hard thing to do, especially globally, right? Because we may be able to regulate, who receives gene editing in the United States, but we might not be able to regulate this all across the world. And this can give people who are wealthy an advantage over others, right? Because wealthy people will have access to this new technology, and they can get around regulations potentially by having enough money to go to a country where the regulations on gene editing are a lot less strict. For example, somewhere like China. And so there have been 2 twins born in China that have had their genome edited by CRISPR. The gene that they had edited out is the receptor for HIV virus, and so these babies can theoretically never get HIV, but this was done very prematurely and with a lot of disagreement by the majority of the scientific community, and there have not been any humans edited